Let's keep the ball rolling with our next speaker, Dr. Iraola. Feel free to share the slides while I introduce you. So Dr. Iraola is a group leader of microbiology genomic laboratory at the Institut Pasteur de Montevideo in Uruguay, and adjunct professor at the Universidad Mayor Santiago de Chile, as well as an affiliate at the Wellcome Sanger Institute. He is passionate about translating scientific advances into solutions that help sustainable human development while reducing global inequalities. To this end, Dr. Iraola has developed and coordinated several large-scale national and international projects, such as the Latinbiota project, and deploying the first real-time COVID-19 genomic surveillance program in Uruguay, just to name a few. So please take it away, Dr. Iraola, and we'll be happy to hear what you have to speak to us.
Okay, thank you. Are you seeing my screen now?
Yes, looks great, and we can hear you as well.
Right. Thank you for the invitation and for the introduction as well. So I will be talking about our work that has been focused in the last 2 years in unlocking and understanding the composition and structure of the human gut microbiome in underrepresented populations, and in particular in Latin America. And this is motivated by the fact that we all know that the genetic and taxonomic diversity of the gut microbiome is still unsaturated. That means that every new population or every new group of people or cohort, when we analyze them, is composed by newly discovered species that we didn't know about the existence before. So this is particularly relevant, and sorry, because these first slides are going to be a bit overlap with the previous talk from Dr. Poyet, but I think it's worth mentioning that today the availability of metagenomic data, in particular from the human gut, is extremely biased towards the northern hemisphere, and in particular to high-income countries. And this leaves many entire regions of the world, like those that are highlighted there: Latin America, Africa, Eastern Europe, Middle East, Southeast Asia, as black holes of information regarding the human microbiome. And of course, this impacts our understanding of the role of the human gut microbiome in health and disease. With the aim of contributing to mitigate this bias, we started our project, and after that, Dr. Poyet showed some figures that were published in this very nice paper that indeed highlighted this preliminary results we had from the screening of public data sets. We put our emphasis on Latin America, of course, because it's the continent in which I got born, and I am from Uruguay, and I developed my science here. But Latin America itself is a very interesting continent, because it's a kind of chrysalis of different cultures, different levels of urbanization, different socioeconomic levels of different countries. We have had different waves of immigrations across the year. We have a Western lifestyle, but there are still, for example, indigenous communities in many countries, and it's very vast in different aspects. But also from the health point of view, we have a high prevalence of many inflammatory and non-transmissible diseases like cancer, and particularly obesity. Latin America has one of the highest obesity rates in the world, and this is going to make sense at the end when you see some of our results, that I want to mention that when I was seeing the results from Dr. Poirier previously, I was very excited because the results you are going to see in my talk, I promise, were not premeditated, but they match a lot with what was shown previously. So for studying the Latin American microbiome, I created what I call the Latinbiota Consortium, that today is composed by more than 10 countries. Here I am highlighting the institutions and the countries that originally conformed the consortium. And these are 8 countries: Mexico, Colombia, Ecuador, Brazil, Bolivia, Chile, Argentina, and Uruguay. And from all these countries, we managed to gather information and samples in the beginning. So the phase 1 of our work was from 2019 to 2021, in which we created a multi-kingdom catalog of the variation of human gut microbiome in populations from these countries. We managed to collect over 600 samples from 8 different countries, and most of these samples were from healthy adults. We also got samples from urban lifestyle and rural lifestyle, and also in very specific cases, we got samples from some native isolated communities from some countries. So we perform shotgun metagenomics over these samples, and we analyze them using a genome-centered approach, building assemblies and mags from viruses and from bacteria mainly. So focusing on the bacteria, with this data set, we improved the quality of hundreds of previously described bacteria from the human microbiome, but also we discover an interesting number of new species, new bacterial species that didn't match to any previously known bacterial species inhabiting the human microbiome. So to understand the differences and the prevalence of these newly described bacteria, we compare our Latin American dataset with a global dataset consisting in over 30,000 gut metagenomes. And focusing on this new bacteria, there are more than 300 bacterial species discovered in Latinbiota. In panel A, you can see the distribution of these bacteria across the phylogeny of known bacteria from the human gut. But was more interesting for us was to understand the prevalence of these newly assembled and discovered bacteria from Latin America in other continents. And we found that the highest prevalence of these 300 novel bacteria was in Africa. So we uncovered this connection between the African and Latin American microbiome, and we hypothesized that this could be related with the lifestyle and with the less industrialized lifestyle that is a general pattern in these 2 continents. And when we analyze the taxonomic assignment of this newly discovered bacteria, we found that the first and top taxa represented among them was the order Ocelospirales. And this order Ocelospirales also, when we calculated the prevalence of the Ocelospirales in the different regions, we found then that they follow a vanish-like pattern, in which these species in particular are more prevalent in less industrialized population and vanish from more industrialized populations, mainly from high-income countries. So we decided to explore a bit more the population structure of the Oscillospiralis order and the Oscillospiraceae family in particular. Indeed, these plots are from the Oscillospiraceae family. So we perform a population structure analysis and compare the functions that were enriched in those lineages that were more prevalent in Latin America with respect to the rest of the world. And one thing that was interesting for us in this moment was the defense mechanisms that were mainly composed by AMR genes. Most genes that were more abundant in Latin American Oscillospiralis or Oscillospiraceae, sorry, were AMR genes. So we perform a more in-depth characterization of the AMR genes across the whole microbiome. And we found that Latin America is the region with the highest prevalence of AMR genes in the gut microbiota. And most of these genes are from the beta-lactamic and tetracycline classes. That also makes sense with the antibiotics that are mainly used for human treatment. Also, many species, including many of them from the Oscillospirales order and Oscillospiraceae family, were significantly associated with the rural-urban lifestyle within Latin America, and this can be shown in this plot. So we started to uncover some relationship between the lifestyle, the composition of the human microbiome at the global and continental level, and also other important features like antimicrobial resistance and potential reservoirs for antimicrobial resistance. But also, as I mentioned, we focused on the viral species, and here I am basically talking about phages, and comparing against the current databases of phage species that are present in the gut microbiome. We uncover over 9,000 new viral species from Latinbiota from this dataset. We also show this strong relationship or correlation between the viral diversity and the bacterial diversity. That, of course, makes sense because there is some ecological equilibrium between the phages and their hosts that are bacteria. But interestingly, among those bacterial hosts that were mostly represented among the newly discovered phages, we detected the Oscillospiraceae. So that means that many new phage species that are being detected in Latin America are infecting this Oscillospiraceae family that seems to be very important for any reason in the normal gut microbiome from Latin Americans. Indeed, it is one of the taxa that is mostly associated to the Latin American region if you perform an association analysis based on the abundance of these bacteria in our guts. So this is more on the viruses. There are different taxa and different species of viruses, but the main point here is the Oscillospiraceae as one of the main hosts for these new viruses. Also, we did a bit more on this and compare the the prediction power of the viral and versus the bacterial component of the microbiota in terms of geographical origin. So we also uncovered that the viral composition of the microbiome has a more powerful signal to predict the geographic origin of a sample at the regional, but also at the country level. So this is something that should be explored more in depth, but is important also from the biomarker discovery point of view. And finally, I wanted to share with you some results about the overall coding and functional potential that is coded by genes present in the microbiota of Latin Americans. And here, basically, we uncover over 200,000 new protein clusters that are new proteins at the 90% level of identity with any previously known protein. And 30,000 of these 200,000 are completely of unknown function. We couldn't annotate anything, any clue about the function of this protein. So there is a vast genetic reservoir in there to be explored and to define which functions, molecular or biological functions, these enzymes, these proteins could be playing. So in conclusion, the phase 1 of Latinbiota contributed to the discovery of over 300 new bacteria and 9,000 new viral species. Novel species are mostly related to the Oscillospiralis and Oscillospiraceae taxa. We also, here there are some highlights about some results I didn't mention due to time constraints, but we developed a new bioinformatic method for detection of viral species with higher accuracy. We uncover for first time the presence of some phage lineages in Latin America. Also, we contributed to enhance the quality of reference gut bacterial genome, some reference genomes from gut bacteria in public datasets. We also showed the possibility of predicting the origin of a sample from gut metagenomic data, and the virome here is the more informative component of the microbiota. And also the results I just mentioned about the proteins, the new proteins in there to be understood, and also the highest prevalence of AMR genes is something that caught our attention, and is something that we will keep exploring in the future. What's coming next? Well, we have just got some funds from the Pasteur Network to expand the concept we developed in Latinbiota to other underrepresented regions. And in the next few years, we will be working in particular with countries in Northern Africa and in Eastern Europe to gather information about the microbiome in these populations, with particular interest in the role of these populations as reservoirs of antimicrobial resistant genes. Also, we are expanding Latinbiota. We have a second phase in which we want to expand the geographic, ethical, and lifestyle origins of the samples. We are welcoming new collaborators, and we are very excited also to start collaborating or discussing with the people that is today presenting and participating in this incredible symposium. We also plan to explore other body sites. We are very interested in the oral microbiome, also collecting saliva and fecal material from the same individuals, and also integrate our metagenomic data with other sequencing technologies that can improve the resolution at the strain level, or the recovery of mobile genetic elements using this. This is going to be using long reads and other approaches like proximity ligation sequencing. And of course, now we have a baseline of variability of the microbiota at the continental level in healthy population. And we now we want to start analyzing what is happening with different disease cohorts that are very important for the countries that are conforming the Latinbiota Consortium, and those diseases that are important at the continental level. And that's it. This is my group at the Pasteur Institute, and this at the right are some of the people from Sanger that has been helping a lot during these years in every step of Latinbiota. And these are my contacts and social networks. And so thank you very much.